All right, now reactions don't typically occur haphazardly in healthy cells. They're going to occur sequentially, so that way the products of an early reaction will become the reactants for another reaction. This is what's going to be known as a metabolic pathway. A metabolic pathway is going to begin with a particular reactant, proceed through several intermediates, and terminate with a particular end product. So we're going to start off, we're going to convert A to B. A is going to be our initial reactant or our initial substrate. Once we convert A to B, B can be converted to C, C can be converted to D, D converted to E, and F converted to E converted to F, and G converted to F converted to G. G will be our final product. This whole process is what's known as the metabolic pathway, and it may seem a little bit out there, but don't worry, we're going to spend a lot of time talking about metabolic pathways and enzymes. Now, the interesting thing about these metabolic pathways is that, as we've sort of already mentioned, a lot of reactions do not occur spontaneously. They're going to require a little bit of help. And even the reactions that do occur spontaneously are often going to occur very, very slowly. Typically, we are going to need some sort of reaction or some sort of catalyst to help drive these reactions forward. The method we're going to use to drive these reaction forwards would be enzymes. Enzymes are going to be protein catalysts. They are going to be the reactants of an enzymatically catalyzed reaction are often called substrates. Typically, these enzymes are protein molecules. They're going to function as catalysts, meaning that they're just going to help speed up the chemical reaction. They're going to facilitate chemical reactions. Like I said before, and we will talk about this quite a bit in our discussion group, but when we are talking about chemical reactions, what goes in is a reactant. What comes out is a product. Now, just for the sake of terminology, and I do want to clarify this because you will see it in your labs, when we are talking about an enzymatically controlled reaction, in other words, a reaction that requires an enzyme, we will call what goes in a substrate. What comes out is still a product, but what goes in is a substrate. I'm not super picky about this terminology, but you will see it in the textbook and you will see it in lab, and so I just want you to be aware of what substrate means. Substrate, every year I get students defining substrate as like the basal layer of a tissue. It's not what we're talking about here. A substrate in this case is going to be the reactant of an enzyme-controlled reaction. So the substrates for the first reaction are going to be converted into products. Those products then serve as the substrates for the next enzyme catalyzed reaction. One reaction leads to the next in a highly organized, regulated manner. So each enzyme is going to accelerate a very specific reaction. Enzymes are very, very specific. This enzyme that we have here cannot convert C to D. Enzyme 1 cannot convert A to C, cannot convert B to C. Enzyme 1 has one job. He is going to help A become B, and that's it. That's his only job. Enzyme 2 is going to be there for converting B to C. Enzyme 3 is going to be there for converting C to D. They're very, very specific. And the end product, so each reaction, the metabolic pathway requires a very unique and specific enzyme, which means that if we need D, but enzyme 2 is missing, we're never going to get there. The end product will not be formed unless all the enzymes in the pathways are present and functional. This is where a lot of genetic disorders, especially genetic metabolic disorders, come from. There is one protein that is missing, or one protein that is slightly malformed or has a mutation. If that one protein is missing, it doesn't matter if all the other enzymes are healthy. It doesn't matter how much A you put into your body, you're never going to get to D if enzyme 2 is missing. So this is where it's super important to have all of the enzymes necessary for a metabolic pathway. So we're going to talk about the enzyme substrate complex. How does enzyme 1 convert A into B? The enzyme is going to convert A to B by using an enzyme substrate complex. In other words, what's going to happen, only a small part of the enzyme is going to interact with the substrates. This is what's known as the active site. It's going to cause, once the substrates bind to the active site, it's going to cause the active site to change shape. The change in the shape is going to force the substrates together, initiating bond formation. Once they're sort of bonded together, they're going to snap together. It's going to put those pieces together and force them into the end product. Now, this is a bit abstract. I'm sorry we're doing the talk this way. I normally do a chalk talk with lots of terrible, terrible drawings at this stage. So please join me for a discussion group, and I will be happy to draw my terrible drawings out for you to explain active sites, substrates, and enzymes. But you can kind of think of it as a box of Legos. If you want to snap two pieces together, you can just sit there. I mean, two pieces are gonna go together, but the problem is that they're not gonna just go there on their own. Even if you add energy to the mix, like if you start shaking and shaking and shaking and shaking your box of Legos, the odds that you're going to just be able to shake that box of Legos until two pieces snap together are really low. It's possible, but it's going to take a lot of time and a lot of shaking. Instead, what you could do is you could use your two hands. Your two hands could be the enzyme. Your two hands can pick up the two pieces that you want to snap together and then physically push them together until that connection is formed. You physically push them together until a covalent bond forms, until they snap together in place, and now you suddenly have two pieces you suddenly have one piece where you once had two pieces. You put those two pieces together, you've got one piece. We're going to talk about, this is going to initiate our bond formation, is when we force those two substrates together. Now, the induced fit model of the active site is when the enzyme is going to be induced to undergo a slight alteration to achieve optimum fit for the substrates. And again, we will talk about all of this in our discussion group because, boy, I can do a lot better job when I can draw you some terrible drawings of what this looks like. I don't have the interactive technical ability to make this enzyme do that for me. So we're going to look at how this enzyme substrate complex works. And we're going to start off by looking at, I'm going to skip ahead and we're going to look at a synthesis reaction because that kind of fits with what we've already talked about. So enzymes are going to be used for a synthesis reaction. What happens is we start off with two individual substrates, we bind them to the active site of the enzyme, and the enzyme substrate complex is going to snap those two pieces together and release them in the form of a product. So we're going to take two substrates, we're going to put them into the enzyme, take these two substrates, put them into the enzyme, snap those two pieces together, and then we're going to release the product and we have our enzyme. So this is how enzymes can start a synthesis reaction. Substrates are going to be joined together and released as a single product molecule. Now, just as you can take that product, you could take those two pieces that are stuck together and you could shake and shake and shake. It's going to be a long time before they spontaneously break apart. A little bit of energy is going to be required to break those two molecules apart. And we can do this through a degradation reaction by using a similar enzyme. An enzyme can not only can we use enzymes to put products together, or we can put substrates together to form products, we can also take a single substrate and we can put it into that enzyme and snap them apart. So just like enzymes can be used to snap products together or snap substrates together, it can also be used to snap substrates apart. So we can put that substrate into the active site of the enzyme. We're going to snap those two pieces apart and we're going to release two products and our enzyme. Now, one thing I want to point out is that through this entire process, whether we are doing a synthesis or a degradation reaction, the enzyme, look at this enzyme. Is he changed at all in this process? No. Is he part of the end product? He is in no way. The substrate goes in, the product comes out, the enzyme goes on his merry way. He's not altered by this process. He's not used up by this process. Once he releases those products, that enzyme can go on to catalyze this reaction over and over and over again. So we're going to put a lot of that information now into words so that way you can have all those notes written down. So we're going to talk a little bit about activation energy, or the process of using energy to drive these reactions forwards. The reason why we need, or we're going to talk about the reason why we need enzymes to do this. And as I kind of already said with our Lego analogy, those Lego pieces are not going to snap together by themselves. A little bit of energy is going to be required to start that reaction. So molecules are very rarely going to react with one another unless they are activated in some way. Those Lego pieces are not going to snap together on their own. Energy is going to have to be added to at least one reactant in order to initiate that reaction. Even if it's ultimately going to be an endergonic reaction, even if we're breaking a molecule apart and we release energy in the process, we're releasing energy, but still a little bit of energy is going to have to be invested first. We're going to have to put a little bit of energy in order to get smooth sailing after that. So energy is going to have to be added to at least one reactant to initiate that reaction. The energy that you start off with, or that you have to throw in the pile to get it going, this is called activation energy, or EA, the energy of activation. I think your textbook refers to it as the energy of activation. To me, that's a little bit wordy, and I've always called it activation energy. Every textbook I've ever seen before calls it activation energy. You can use either term, it means the same thing. It's the amount of energy that you have to put in to start the process. So how do enzymes work? Enzymes work by lowering the activation energy. They bring substrates into contact with one another, which increases the rate of the reaction because we lower the amount of energy required. Great example of this that I like to use. I like to think of an enzyme as a dating app. When I was looking forward to meeting my husband, I moved to a new city, and I was like, how do I go about meeting new people? I want to date. I want to meet people. I want to get married. I know what I'm looking for. How am I going to find people? Now, I could either get dressed up every single night, go out, I don't know, clubs, bars, health clubs, I don't know where people meet people. Maybe you just troll around the grocery store, hoping that I randomly bump into Mr. Right. That's a lot of energy. Or what I can do is use a dating app. A dating app is going to lower the activation energy, and a dating app is going to bring two people together. You see if you fit. If you fit, you go off, you be a couple, and you're done. So I can either go up and put in a lot of energy, hoping to randomly bump into Mr. Right, or I can use a dating app to bring Mr. Right to me while I'm sitting on my couch. See, now, when there's still energy going to be required. I'm still going to have to put energy into this. I still have to create an online dating profile. I still have to meet with the people. I still have to talk to the people on the app. I still have to meet people on the app. But now, instead of, putting on makeup and getting dressed and leaving my house and going out every single night, I only have to do it 2 or 3 times. I only have to do it with the people I'm truly interested in meeting. So it just lowers the amount of energy that is needed to find somebody. So there you go. That's my dating app analogy. Now, here's the interesting thing about the dating app. Is the dating app used up when my husband and I found each other and we started dating and we went off on our merry way? Does the dating app still exist? Yep. It's still there. Is it still bringing couples together and sending them on their merry way? Yep. It's not used up. It's not affected. It's not in any way destroyed by the reaction. It's not part of the reaction. I met my husband. We started dating. Was the dating app still part of our lives? No. Hopefully it should not be. Once your substrates get brought together into a product and go off on their merry way, hopefully the enzyme is not still involved in that process, or you're going to have some issues. No, the enzyme is no longer part of the reaction. All it does is drive the reaction forward. All it does is lower the amount of energy that it takes for that reaction to happen. So even if it's a spontaneous reaction, even if there's more energy at the beginning and we're going to release a whole bunch of energy in the process. So our energy of our reactant is higher to begin with and lower to begin with. It was a lot more work for me to be single. I'm a lot happier in my couple. So to me, this maintaining my couplehood, my relationship is less energy. So in order to get there, though, I had to put in a little bit of energy. Had to put in a little bit of energy to make that happen. Without the dating app, that's a whole lot of energy. That's a whole lot of dressing up and meeting people and hoping to bump into Mr. Right. With the dating app, it's a little bit less energy. Still requires energy. Still going to require the input of energy to drive that forward. But the activation energy is going to be less. So, there are going to be certain things, so that's our enzymes. Now, please let me know if you have any questions about how enzymes work, or and if you take issue with my dating app analogy, that's fine. Use another analogy, something else that makes sense to you. I just use that because it makes sense to me. But yeah, we're going to pick up this lecture in just a minute with factors affecting enzymes.